Could you go ahead to share your screen? We can just
Yeah.
test whether,
Okay.
it goes smoothly.
Let's see. You see that?
Yes.
Yes. Yeah. All good.
Okay. All right, great. So,
So Julie, I think we can start.
Can start. Okay.
Yeah, you can go ahead.
Okay. Good morning to the friend in China and good evening to the friend in the United States. Welcome to the 234th online forum of Metabolic Age Talk. My name is Julie Bai. I am assistant professor at UT Health Houston. And together with Dr. Liheng Wang from Peking University, today we are going to host today's online seminar. It's our really great honor to introduce today's distinguished metabolic age talk seminar speaker, Dr. Alan Sotil. Alan is the most handsome gentleman and in the metabolic field. He is a distinguished professor, and Dr. Mariam Ahmed Endowed Chair in Metabolic Health at UCSD School of Medicine, where he directs the Institute for Diabetes and Metabolic Health and co-leads the UCLA Diabetes Research Center. He earned a zoology bachelor's degree from Duke University and a PhD degree in biochemistry from University of North Carolina, and trained in molecular biology at Wellcome Research Labs under Dr. Pedro Casas. Previously, Alan directed the University of Michigan's Life Science Institute and held senior roles at Park Davis, now is Pfizer. Alan is an elected member of the National Academy of Medicine and the fellow of AAAS, ASPET, and ASCI. He has received numerous honors, including the Rosalind Yellow Award, John Jacob Abel Award, Goodman and Gilman Award. Alan is a pioneer in the insulin signaling and metabolic diseases. His research explored the hormonal regulation of lipid metabolism, inflammation, glucose homeostasis, and therapies for metabolic dysfunction, associated hepatitis, and many other metabolic diseases. His work, supported by NIH grants, has transformative, understanding of the cellular signaling in diabetes, cancer, cardiovascular disease. He has over 300 publications and holds 19 patents and serves on the advisory council, grant review panels, and editorial boards of many leading journals such as Cell Metabolism, Science, and he also published, many high-rank journals, including the Science, Translational Medicine, Cell Metabolism, Science, JCI, and Nature Medicine, etc. With extensive, FDA experience in drug development, his contribution bridge the academia and industry, driving innovations in the metabolic health. Beyond his scientific achievements, Dr. Sotil is a great mentor who has trained many, prodigious investigators in metabolic research field, also including, our today's audience and panelists. For example, Dr. Jun Liu, who is a professor of Mayo Clinic, and Dr. Xiaowei Chen, who is a professor at Peking University, and Dr. Peng Zhao at UT Health San Antonio, and many others. Today, we are also honored to have several panelists to lead our discussion after Alan's presentation, including Dr. Peng Zhao, who is Alan's previous postdoc, now is become independent and research is an assistant professor at UT Health San Antonio, whose research focuses on the investigating the regulation of energy homeostasis, lipid metabolism, inflammation, and cell death in the pathogenesis of diabetes, liver disease, including MASH and HCC. And also we have Dr. Aaron N., who is an assistant professor at UT Health Houston, who's my colleague. He's studying the adipose tissue, adipose mitochondrial impact on the metabolic homeostasis. And also we have Dr. Hannah Xiaoyan Hui, who is the assistant professor at Chinese University of Hong Kong. The research interest of Dr. Hui lies on the adipose tissue, particularly investigating the molecular basis of adipose tissue remodeling and its physiological relevance in obesity and cardiometabolic diseases. And we also have Dr. Meng Xijiang, who is a professor at Capital Medical University in China. Her research interests focus on the preclinical evaluation and pharmacological management of metabolic syndromes. And also we have Dr. Hongxue Shi, now who is an associate research scientist at Columbia University in Irvine Medical Center. Hongxue now recently received his K99 award, and he will start his new position as an assistant professor at Louisiana State University Health Science Center this July. Congrats, Hongxue. And his work mainly focuses on the investigating the role of macrophage afrocytosis in the development of MASLD and MASH. Please join me in giving a warm welcome to Dr. Alan Saltiel. The title of his talks today is Responding to Metabolic Stress. Welcome, Alan.
Okay, well, thank you very much. It's really a pleasure and a great honor for me to be here, and also to see many of my old friends and colleagues, and like Pong and Xiaowei, and if June is there somewhere, and others who are really responsible for a lot of the basis for a lot of the stuff that I'm going to talk about today. By the way, can you guys hear me okay? Is the sound good? Okay.
Yes, sounds good. Yeah.
Yes.
Okay. So I'm going to start today. For those of you who know me, it'll be a little bit boring, but for those of you who don't know me, just by reminding you that my laboratory has been interested in one overarching question, really, since I started it many, many years ago. I'm too embarrassed to tell you how many years ago, but many, many years ago when I started my lab. And that was really, how do metabolically active cells, especially fat cells and liver cells, decide whether to take up and store energy or burn it or release it for other cells to burn in response to hormonal or nutritional cues that arise during fasting and feeding. So this is the main question that we're interested in. And then layered on top of this question is, how do the signaling pathways that control metabolism adapt to conditions of metabolic stress? And when I say a metabolic stress, I mean both catabolic stress, like starvation or exposure to cold in animals, or anabolic stress, like overnutrition or obesity. So these are the questions that we're really interested in, and we've been trying to understand the basis for this and the mechanisms that underlie this adaptation of cells. And I'll cut right to the punchline and tell you that one thing, a lesson, I guess I would say, that we've learned over all these years as we ask this question about adaptation to metabolic stress, is that irrespective of the kind of stress, cells always end up responding in the favor of storing energy. So they respond to both starvation and they respond to obesity, and they respond to cold by ultimately finding a way to adapt by storing more energy. And the reason they do this is because they want to be able to survive the famine that's coming up. So they find a way to tweak the cell, and this is especially true in the fat cell. They find a way to store energy and repress energy expenditure. So we've been thinking about this, and this is what I want to talk about today. So I'm going to focus my comments today mostly on the anabolic stress side of the equation. So we're very interested in what happens in starvation and cold to allow cells to preserve energy expenditure. But I'm going to talk today mostly about the other side of the equation. That's the overnutrition or obesity side of the question, where cells are really geared towards adapting to be able to store more energy in the long run so they can survive the famine that's coming up next. And we can model this, of course, in the black 6 mouse, because as many of you know, the black 6 mouse is a pretty good model for humans, at least for some humans. And we like it. We like the black 6 mouse because when we put them on a high fat, high calorie diet, they predictably gain weight. And as they gain weight, we can measure all kinds of stuff in these mice. Among the things that we can measure is we can put them in metabolic cages and measure energy expenditure. And what we see is something that I'm sure many of you have noticed before. And that is when we first put them on this high calorie diet, high fat, high calorie diet, they respond by compensating for all these calories by increasing energy expenditure. But that lasts for only a certain amount of time. They hit a peak, and then energy expenditure starts to decline. And if you go out for several months, you can see that sometimes energy expenditure is actually below where it was when we started. And in my lab, this was first really pointed out by Peng Zhao when he first showed this. And this change in reducing energy expenditure, we can look at in the fat cell, is accompanied by signaling pathways, anabolic signaling pathways, like the mTOR pathway, that are activated constitutively. When we look at catabolic pathways, like the pKa pathway inducing UCP1 or phosphorylating HSL, or the AMPK pathway, the two major catabolic pathways in fat cells, these are repressed. So we're really interested in trying to get to the bottom of what's the mechanism that causes this. But the basic line is that there's more energy storage and less energy expenditure in this kind of anabolic stress and metabolic stress. So how's this happening? So again, today we've been working very hard on this in liver, and very small amount of our work has been done in muscle, but a lot of our work has been done in adipocytes. So we've been trying to understand what happens in the adipocyte. And one way in which the adipocytes adapts to metabolic stress is through mounting an inflammatory response. So this is something I'm not going to talk about today, but we know that inflammation is triggered in response to overnutrition and high-fat diet. And work that we started in my lab was started about in the early 2000s. Carrie Lemang in my lab was the first person to look at this. And over the years, what we've learned is that we think the major primary action, or the way that inflammation is primarily exerted once it is triggered in the fat cell, is to repress energy expenditure. So we've learned, for example, that catecholamine insensitivity or catecholamine resistance is a consequence of inflammation through downregulation of beta-adrenergic receptors that we published a few years ago through activation of the phosphodiesterase pathway that degrades cyclic AMP. We know that AMPK is repressed in response to inflammation. This is Peng's work that was published a few years ago in Cell through direct phosphorylation. We know there are changes in glycogen metabolism. We do a lot of work on glycogen, and we have a paper coming out in the JCI, I think in a few days about this, mostly liver in this case. We've done a lot of work on glycogen I'm not going to talk about today. We know that triglyceride synthesis is increased in inflammation in liver and fat mainly due to the reesterification of fatty acids. We had a paper on this a couple of years ago, and we know fatty acid oxidation is also reduced in response to inflammation. Work from Shannon Riley when she was in my lab. So there's lots of things going on with inflammation. that are triggered by the metabolic stress that the fat cell sees. And there's lots of stories here. And I'm showing you all this, but I'm not taking responsibility for these today because I'm not going to talk about this. I'm going to tell you about another mechanism that we've bumped into that's not a direct consequence of inflammation. At least we don't think so. Now, this is a story that is a kind of an interesting story. It's a long, winding story that began that we kind of bumped into accidentally, and it started from work that we were doing to understand something about insulin action. And I really want to tell you the story today because I think it's a great lesson for some of the younger people and students in the audience that we don't always find what we're looking to find. And sometimes we bump into things that are a little bit unexpected. And when that happens, we've got to be ready to take advantage, even though these discoveries are sometimes surprising to us and sometimes don't make us all that happy when we first see them. It's really important to pursue them and try to figure out what's going on. So this story started many years ago in our attempts to understand how insulin controls glucose transport in fat and muscle cells. So I think most of you know that glucose transport that's increased by insulin is increased due to changes in the protein GLUT4, the glucose transporter GLUT4, which is regulated by insulin by virtue of
哎，我走退了
its localization in cells.
哎
You guys hear me okay?
，sorry，
There you go. So we've been trying to understand the mechanism by which the glucose transporters are recycled in response to insulin. And I'm not going to spend a lot of time talking about this today, but I guess about 15 years ago, those of us who were working in this field, which included a lot of people then, hardly anybody now, I may be one of the few people left who's interested in this question, but we started to focus on the role of small G proteins that resided at the intersection of signaling and trafficking of the protein. And over time, we've worked on just about all of these proteins that are outlined in this cell. And the one that was particularly interesting that we bumped into in the early 2000s was the Rho family protein TC10, which we showed was activated by insulin. But this is the insulin receptor, which activates TC10. So when we discovered this G protein, this is the Rho family protein, also called RhoQ. We were very interested in trying to understand what it was doing, and since it was a G protein, to try to understand what its effectors were. So we took advantage of this technique that we've been doing in my lab back in the last millennium that a lot of you probably have never heard of because it was this ancient technique that was very powerful at the time called 2-hybrid screening. And we use this 2-hybrid screening to find effectors of TC10 that only bound to active TC10. So with this, we made this 2-hybrid library from adipocytes. That was a really great library. It was made in the early 90s. And we used a mutant form of TC10 that was constitutively active as bait with a constitutively dead form as a control. And we pulled out a bunch of interesting genes that encoded proteins that bound to TC10. And among these was one that was really interesting, and that was a protein called Exo70. Now, Exo70 turned out to be a member of this 8-member complex of proteins called the exocyst, which had been studied in yeast primarily by my new colleague now, Peter Novick, who works at UCSD. I was still at Michigan when we bumped into this. And what Peter had shown that this exocyst complex, this 8-member complex, assembles at the plasma membrane in yeast to form a targeting site for the vesicles that are undergoing exocytosis. When a G protein in the plasma membrane in yeast, in this case it's Rho3, is activated, this results in the assembly of the exocyst. And then it's got to be found by the vesicle, and this happens to another G protein. In yeast, that's the G protein Sec4. So when Sec4 is active, it can bind to other members of the exocyst. And now it traffics across an actin cable to find its way to the plasma membrane for targeted exocytosis. So we were excited about this when we first saw this because we thought, maybe TC10 was doing something like this in mammalian cells, especially in fat cells. And we had a paper on this in Nature in the early 2000s, a work from Mayumi Inoue and others in my lab. And what we showed was that TC10 is activated by insulin. It binds to exo70 to assemble this exocyst, which sets up a targeting site for vesicles as they move towards the plasma membrane. And then we wanted to try to figure out how the vesicle recognizes this exocyst. And Xiaowei Chen then came into my lab first as a rotation student when he worked with Jun Liu. And Jun Liu somehow was able to tolerate Xiaowei. They didn't cause too many problems. So Xiaowei came to the lab as a graduate student, and Xiaowei showed something very important, and that was the discovery that RAL-A resided in the GLUT4 in the vesicle and was responsible for the recognition of the exocyst to cause the ability of the GLUT4 vesicle to find the plasma membrane for targeted identification and docking and fusion. And just to show you one experiment that he did, of the many experiments that he did, he transfected fat cells with a GLUT4 promoter, a GLUT4 reporter gene that had a MIC tag. And you can see that in these cells, if he treated these cells with insulin, he caused a translocation of GLUT4 to the plasma membrane. And then he used this new siRNA technique that was just available at that time to knock down RAL-A, and he showed that this blocks the effect of insulin. So RAL-A was really responsible and required for insulin action in this process. So Xiaowei, I'm reducing his work to one slide basically, but Xiaowei showed that RAL-A is important in insulin action, that it was activated by insulin through a mechanism that required AKT. He was interested in finding out how that worked, and he turned out that it was due to the inactivation of a cognate gap complex that Xiaowei was able to clone and characterize. This gap complex had two subunits that we called at the time RGC for RAL-GAP complex. Now they're known as RAL-GAP B, which is a scaffolding protein required for the stability of this complex, and RAL-GAP A, which is a catalytic gap protein he showed had gap activity. And he also showed that AKT could modify this protein, and this resulted in the inactivation of the gap. So I'm summarizing his entire graduate career in 1 minute. I'm sure he doesn't appreciate that, but what Xiaowei was and Darolito and others in the lab were able to show that insulin through activation of AKT would phosphorylate RAL-GAP. This resulted in the sequestration of the protein, and this led to the activation of RAL, which can then interact with members of the exocyst. And it's way more complicated than I'm letting on. There's a lot more going on in this process. If you're interested in learning about this, here's a big bunch of papers that took us 15 years to really characterize and understand this process. I think today, if we made this discovery, all this would have been in one paper probably, but back in the day, we were able to fragment it into different papers. So I really want to show you this to set the stage for what we did next. And we were very interested in validating the role of this RAL pathway in the regulation of glucose homeostasis in vivo. So we took 3 approaches, and 2 of these I want to talk about today. One was to introduce a gain-of-function mutation in mice, in this case, to conditionally knock out the RAL-GAP, which resulted in the constitutive activation of RAL. And then we took the opposite approach to use RAL inhibitors, which I'm not going to talk much about today, or a genetic approach to create a loss-of-function situation in which we conditionally knocked out RAL-A itself in fat cells. We've done this in muscle and fat and liver, but today I'm just going to talk about the work that we've done in fat cells. And this last thing was the work of Wenmin Xie, and I'll get to her work in a minute. So the first thing we did was to knock out RAL-GAP, and that so in this case we knocked out RAL-GAP B conditionally in fat cells. We made flox mice, crossed these with the Dippo-Q mice. As you can see here, we're able to knock out RAL-GAP B, which is a scaffolding protein required for the activity of RAL. both in IWAT and EWAT and brown fat, and no other tissues. And as you can see, when we did a pull-down assay looking for activation of RAL, and this is an assay in which we immobilized one of the effectors of RAL and just pulled it down. So it only binds to GTP bound active RAL. We saw a very substantial activation of RAL in these cells. Much more activation, in fact, than you see if you just give cells insulin. So the GAP protein really is the primary controller of the activity state of RAL. So we took these mice and looked at glucose uptake in these mice. And so we look here at wild type and knockout mice. This is just at the basal level, looking at glucose uptake. And first of all, we were surprised that there was no effect, despite the fact that we see very substantial activation of RAL in IWAT and EWAT, no effect on glucose uptake in these cells, but a major increase in glucose uptake, even in the basal state, in brown fat. And in muscle and heart, there was actually a reduction in glucose uptake. And this was not that surprising because all the glucose was going into brown fat. So we decided to look at this in more detail. We did a euglycemic hyperinsulinemic clamp study. I think most of you are aware of this. You hold the glucose levels constantly. You increase the insulin levels to maximum levels, and then you modulate the glucose to keep it constant. So it's a way to do a number of things, but among these is to look at the glucose uptake in different tissues with tracers. And first, I want to point out what happened in just wild type mice when we looked at fasting conditions and clamp conditions. So you can see, if you start on the right side, insulin, so hyperinsulinemic clamp, so these are maximal levels of insulin, have a big effect on glucose uptake in heart and skeletal muscle, a big effect in brown fat, and really does almost nothing to white fat depots. Either IWAT, there's a small effect, and EWAT, there's really hardly any effect at all. So I have to say that this was not what I was expecting because, up to this point, for all these years, we've been working on glucose uptake mechanisms. We've been looking at what we thought were a model of white fat. But the truth is that insulin really doesn't do much to glucose uptake in white fat in vivo. And we can come back and talk about this later. Nevertheless, there's a very big effect in brown fat. Now, when we looked at the knockout mice, what we saw was, again, in white fat, in IWAT and EWAT, knocking out RAL gap, despite the fact that RAL itself is really activated, really didn't do much at all to increasing the glucose uptake in white fat. In muscle and heart, nothing on the insulin stimulated side. And again, remember, insulin is manipulated to be really high in these cells, but there was a reduction in basal glucose uptake, which I'll come back to in a minute. But with respect to brown fat, where we're also constitutively activating RAL, even in the absence of insulin, there's a really big increase in glucose uptake and a further increase in the presence of insulin. So this was, again, the lack of effect of white fat and the big effect in brown fat was a little bit of a surprise. But we actually started looking, and there was a paper that came out a couple of years later from us. This is a paper from David James, who did a similar experiment, just in wild type mice, looking at hyperinsulinemic clamp conditions. And you can see, again, he saw the same thing we saw: big effect in heart, big effect in brown fat, big effect in muscle, big effect in quadricep muscle, and almost no effect in fat. So anyway, this is a little bit of a surprise, but we think glucose uptake is not really important in vivo in white fat, although it's very important in brown fat. So what about glucose homeostasis in these mice? Well, so then we went back and did all the usual thing, thinking that constitutive activation of RAL in brown fat, even though it's not doing much in white fat, should give us an improvement in glucose homeostasis. And in normal diet mice, it did in fact give us an improvement in glucose homeostasis. Here's a glucose tolerance test, but it was a pretty small effect. And here's looking at insulin levels were reduced. Here's an insulin tolerance test. You can see they're a little bit more sensitive. It's not a big effect, but there's an effect. It's kind of it's not surprising, I guess, when you think about it, because glucose uptake is only going into brown fat. And as I showed you before, there's a little bit of a compensation to reduce the glucose uptake in muscle. And we thought, well, maybe we need to stress these mice to put them on, really give them some anabolic stress to put them on a high-fat diet and see if that would allow us to see a bigger effect of RAL gap knockout of the activation of RAL. And in fact, that was the case. So we put mice on a high-fat diet. You can see here that the RAL gap knockout mice gained as much weight as the wild-type mice, but the glucose tolerance was dramatically improved. Here's a glucose tolerance test. Here's the knockout mice, and here's the wild-type mice. You can see a dramatic improvement, a big-time reduction in insulin levels. So HOMA-IR was dramatically improved. Here's an insulin tolerance test. Again, the sensitivity to insulin was improved. This is looking at insulin levels after starvation. So the constitutive activation of RAL gap in brown fat really did improve the glucose homeostasis. And this was interesting. I'll also tell you that we then made a brown fat specific knockout of RAL gap. So we crossed our flox mice to UCP cream mice, UCP1 cream mice. And the results looking at glucose homeostasis on a high-fat diet were identical to what we saw here with the adipo-Q mice. So all this improvement in glucose homeostasis is due to what's going on in brown fat. Importantly, I think, for me, this validated the RAL pathway as a key controller of glucose transport in brown fat. And a similar study had been done by a Chinese group looking in muscle as well, where they saw a similar result. So muscle and brown fat are important areas in which RAL plays an important role in controlling glucose transport. And then the third message from this was that insulin and RAL-A dependent glucose uptake primarily is happening in brown fat. So the white fat, there was not really an effect in glucose uptake. So it's really all happening in brown fat. But I don't want you to think there was nothing going on in white fat, because in the adipo-Q mice, where we knocked out RAL gap in white fat and increased RAL activity, there was something happening, and that was clear when we did H&E staining of the fat depots. So you can see here in brown fat on the bottom, there was a whitening of brown fat. So all the glucose that was coming into brown fat, not all of it, but a lot of it was being stored as fat. In EWAT, the fat cells were getting a little bit bigger, but in IWAT, the fat cells were getting a lot bigger. Here's the quantitation of all that data. So it looks like, although glucose uptake is not increased in white fat cells, something's happening there, because fat cells are getting bigger and storing more energy when you knock out RAL gap and constitutively activate RAL. So I'm gonna hold on to, ask you to hold on to that. I'm gonna come back to that in a few minutes. Something's going on. So to summarize what I just told you, constitutive activation of RAL-A by knocking out RAL gap improves glucose homeostasis by increasing glucose uptake, specifically in brown fat. But again, I just want you to note that adipocytes in IWAT are enlarged, and this is not due to glucose uptake. Okay, so there's something else going on in these cells. So what about what happens now if we look at the other side of the coin and knock out RAL? So we do a mutation that reduces RAL activity or knocks out completely RAL activity in fat cells. This was again done by crossing flox RAL mice to the adipo-Q mice, anticipating now that glucose homeostasis will get worse. So on a normal diet, it did get worse, but only a little bit worse. Here's a glucose tolerance test. You see a really marginal, it's actually not even a statistical effect on glucose tolerance in normal weight mice. We did an ITT, we see a very small effect, making the mice a little bit worse. These are the RAL knockout mice, get a little bit worse. A few small changes in insulin. Fat cells get a little bit smaller under these conditions, but really on normal diet, we didn't really see much of an effect. So, we saw a similar small effect in normal diet in the RAL gap mice. We thought to ourselves, let's stress them with high fat diet. Maybe it just, changes in the brown fat are not enough to see a big impact on a normal diet. You really need to change muscle. So we didn't really see much. So we put these mice on a high-fat diet, and then we saw really our first big surprise, and that is that glucose tolerance actually got better. And here's a glucose tolerance test. These are the knockout mice. These are the wild-type mice. Here's an insulin tolerance test where there wasn't much of a change. But looking at insulin levels and HOMA-IR, there was a pretty dramatic improvement in these mice. Now, if you're paying attention, I just told you something that's confusing, right? I told you that a gain-of-function mutant makes mice on a high-fat diet, gives mice a pretty dramatic improvement in glucose tolerance. And now I'm telling you that when we did the opposite effect, a loss-of-function model, we also have an improvement in glucose tolerance. So I hope that if you're paying attention, you're confused, right? And I was confused, and Wenmin was confused, and poor Wenmin had to do this experiment many, many times because that's what you do when you get a result that doesn't really make sense. And just to tell you how I felt, just to give you a depiction of how this made me feel, this is an example of how I felt when I saw this data, when I finally believed that this was the data that we had. Okay, so I would say I was, to put it mildly, I was a little bit upset. But how I should have felt is actually like this, because what we were learning from this result was not that our idea was just wrong. What we found that our understanding of the process was incomplete, and that there must be something else going on. Okay, perhaps since we were looking at a G protein, there were some other effectors that we hadn't discovered yet or hadn't considered. So we decided to take a deeper dive into the RAL knockout mice, especially RAL knockout mice challenged with a high-fat diet. And then what we saw allowed us to start understanding this a little bit better, because RAL knockout mice on high-fat diet gained less weight than the wild-type counterparts. It wasn't a huge effect, but it was a substantial effect. And when we saw where the weight loss was, it was all in adipose tissue, and it was mainly in IWEB. So we wanted to dig deeper into this thing. We did a metabolic cage studies in these mice. And if you look at the middle panel here, you can see that there was a pretty significant increase in fuel oxidation and oxygen consumption in the knockout mice compared to the control. No effect in RER. We did the ANOVA two-way analysis, and you can see there was really a pretty big effect in energy expenditure in the knockout mice. So what was the basis of that effect? I'm not sure, but now I want to take you back to the opposite phenotype again, the RAL gap knockout mice, where again, as I showed you before, that these are looking at control and knockout mice, even just on a normal diet. Now you can see that the fat cells are bigger in the knockout mice compared to the control, not just on high-fat diet, but also even on normal diet in this case, where RAL is constitutively activated. And we did a metabolic cage study, in this case, in RAL gap knockout mice on a normal diet, where RAL is constitutively activated. And here you can see we actually did see the opposite phenotype that we saw with the RAL gap knockout mice. There was a reduction in energy expenditure. There's a reduction in lipid oxidation and a reduction in glucose oxidation. So basically, oxygen consumption was generically inhibited in RAL gap knockout mice. These are in the adipo-Q mice. So these are in mice in white. These are effects that are seen primarily in white fat, where glucose uptake is still increased in brown fat. So we wanted to really dig down into the cellular effects, and I should point out that when we did a brown fat knockout of RAL, we didn't see this phenotype. So this is really a white fat specific cell autonomous effect. So we made adipocytes, we made cells that we call ppDivs. These were adipocytes that we differentiated in vitro. We did a seahorse experiment looking at oxygen consumption in these mice. You can see that oxygen consumption by seahorses increased in the RAL knockout mice. We looked at the oxidation of the lipids in these mice, looking at palmitate oxidation. You can see it's increased in these mice. We looked at mitochondrial activity in these mice with a mitotracker stain. We looked at both in primary adipocytes derived from the mice, and we looked at some immortalized adipocytes that Wenmin had made from the primary adipocytes that we were able to study, you can see that the mitochondrial activation in these cells is really increased. Just to make sure that we're looking at an effective RAL, we took those immortalized adipocytes and knocked back in either wild type or constitutively active RAL and looked at oxygen consumption by seahorse and by the mitotracker assay. And you can see that these are the knockout immortalized adipocytes, and this is what happens when we put back in either wild type RAL or constitutively active RAL. We see a repression of oxygen consumption in these mice. So RAL is repressing oxidation of fatty acids in these fat cells. So when RAL is present, oxidation is and present and active, oxidation is reduced. When it's eliminated, oxidation is increased. So RAL is doing something to repress oxidation. So what's the mechanism? So everything we looked at, the fact that there was a real, profound change in both lipid and glucose oxidation in these mice, suggested that the effect was something to do with the activity or the integrity of mitochondria. So when men decided to look at these mitochondria in the adipocytes in this tissue by EM, so she found something, when she did this, she found something really interesting. So these are EM studies that were done on either the wild type or the knockout mice in controlled diet cells where we don't really see a phenotype. And you can see these are the mitochondria in IWAT from these tissues. You can see mitochondria are fairly elongated in these mice. This is a representative of a number of pictures that we took. But interestingly, when we put these mice on high-fat diet, we notice that mitochondria get a lot smaller. So there's a fragmentation of mitochondria in IWAT in wild-type mice. So these are controlled diet, high-fat diet mice and wild-type mice. But when we looked in the knockout mice, we see that the elongated structure of mitochondria are preserved in the face of high-fat diet. So somehow mitochondria are, in the knockout mice, are resistant to the fragmentation that we see in response to high-fat diet. And we could see the same thing in immortalized adipocytes from the wild type of knockout mice. So these are knockout mice. We looked at the mitochondria. We see they're longer compared to the adipocytes from the wild type mice. This is just a quantitation of their data. So we deduce from this that high-fat diet induces the fragmentation of mitochondria. This is something that's been hinted at before, but actually not in fat cells in the literature. So we thought something's going on, and RAL is somehow controlling this process. Most interesting to me was when I said, let's go look at what's happening in visceral fat, in epididymal fat. And it turns out that in epididymal fat, mitochondria are already fragmented, even in the controlled diet, and knockout really didn't have any effect on these cells. So here's controlled diet, high-fat diet. Maybe there's a little bit, the mitochondria are a little bit smaller, but they're primarily already pretty small in this visceral fat, which is already an anabolic tissue. So these experiments told us that there's really a difference in the basic biology of fat cells in IWAT and EWAT. And again, it's not that surprising. Okay, here's again a look these are just normal diet control mice, so wild type mice. And you see the fat. This is just look at fat cells at the same magnification in IWAT, EWAT, and brown fat. You can see they're all really, really different, but these cells are smaller. And then interestingly, when you put mice on a high-fat diet, as I showed you before, you see a whitening of brown fat that we've known about for a long time. You see some enlargement, some plasticity and enlargement in the fat cells in EWAT, in visceral fat. But the big effect is really in IWAT, where these cells take on characteristics that make them very closely resembling what we see in EWAT. They become very much more anabolic. To summarize what I think we're seeing, with respect to metabolism, at least, fat cells in visceral fat are already kind of constitutively anabolic. They have mitochondria that are small and not very active. we look at cells like brown fat, these cells are very metabolically active. Their main job is catabolic. They generate heat. They have a lot of mitochondria. These mitochondria are really big, and they're chock full of uncoupling proteins to generate heat. But subcutaneous white fat, the IWAT cells, are really flexible. They have mitochondria that are bigger than they are in anabolic, not as big and active as they are in brown fat, but they're bigger. They're capable of beta oxidation. They oxidize fat. We had a paper on this a few years ago, actually, with Shannon Riley. And the important thing about these cells is that they're flexible. So in response to metabolic stress, in response to anabolic stress, they undergo the fragmentation of mitochondria, and they now resemble, in obesity, the cells that are found in EWAT. And then again, as you all know, in response to catabolic stress, like cold and activation of beta adrenergic receptors, constitutive, constant activation of sympathetic activation of beta adrenergic receptors, they take on a lot of the properties of brown fat in a process we call Beijing, where among the many things that happen, the mitochondria get bigger and they multiply and they start expressing uncoupling protein to generate heat. So we think this is very interesting that IWAT cells are really plastic. They're really more adaptable and respond to different kinds of stress and metabolism. So this led us to wonder whether RAL activity is being controlled during these processes of metabolic stress. And particularly for this study, is RAL getting activated in IWAT during high-fat diet? And of course, since I'm asking this question, you know the answer. The answer to this is yes. So if we look on the right side, we look at RAL activity with our pull-down assay in mice, in IWAT of mice on normal and high-fat diet. We see that RAL activity is indeed increased in response to high-fat diet. The level of RAL mRNA expression and protein expression, which I don't have here, is also increased. At the same time, the expression of RAL GAP, especially RAL GAP A2, is decreased in response to high-fat diet in mice. And this is also true in humans. So these are studies we've been doing for many years, collaborative studies with Michael Ryden at the Karolinska Institute. And some of you may know this, that he's collected a group of 20 cohorts of patients where they've done RNA-seq and proteomics on adipose tissue and looked at correlation with different of the measures of obesity and insulin resistance and other aspects of diabetes. And as you can see here, levels of RAL GAP A2, mRNA and protein also, are dramatically reduced with respect to obesity. So there's a negative correlation with obesity, looking at things like BMI and HOMA-IR, waist and waist-hip ratio, and circulating insulin. And then interestingly, he looked at a study where they put patients after surgery where people lost weight, looked at the expression of a number of these genes after surgery. These are individual patients, and you can see that RAL GAP A2 levels are increased, meaning RAL activity is reduced after weight loss. So there's a very good correlation with obesity with RAL GAP A2, meaning a correlation with RAL activity. So just to summarize what I've told you about, RAL expression and activity are elevated in obesity, specifically in inguinal fat. Chronic RAL activation is associated with fragmentation of mitochondria and larger IWAT adipocytes. And this is manifest with RAL GAP, and it's also manifest in the RAL GAP knockout mice, even on a normal diet. So you don't even need a high-fat diet if you activate RAL. Mitochondrial fragmentation is prevented in the adipocyte-specific knockout of RAL, and these mice with fragmented mitochondria show reduced fatty acid oxidation and repression of energy expenditure. And of course, this results in more weight gain. And if you block this, you can reduce weight gain even in the face of overnutrition. So what's the mechanism of this? So I'm not going to take a lot of time on this, but since we were looking at fragmentation of mitochondria, we figured it had something to do with mitochondrial dynamics. And as you all know, there are two ways in which the size of mitochondria is controlled, besides autophagy, which was not involved in this process. One was changes in fusion of mitochondria, and the other is change in fission. So you can imagine in obesity, when you see increase in fragmentation of mitochondria, you could have a reduction in fusion or an increase in fission. And to make a long story short, I won't show you the data. We ruled out a role for fusion, and this left us with an idea that fission was possibly increased in obesity. Whoops, sorry. Now, the fission process is controlled mainly by a protein called DRP1. This is a dynamin protein. And this protein is regulated primarily by control of a phosphorylation on two sites. One of these sites is a negative site that's catalyzed by protein kinase A. So when protein kinase A phosphorylates this site, it reduces its activity and it blocks the binding of DRP to mitochondria. When DRP binds to mitochondria, it results in fission and thus the increase in the fragmentation of mitochondria. And when you block activity of DARP through phosphorylation, you reduce this fission. And then in the face of reduced fission, the fusion experiment takes over and mitochondria get elongated. So we wanted to look at the DRP protein and the phosphorylation state of this protein in obesity. So we looked at wild type and RAL knockout mice. We looked at the phosphorylation of the site. We saw it was really elevated in mice in mice in which RAL was knocked out, and this is looking at IWAT. Then we looked at our immortalized adipocytes, looked at phosphorylation on this negative site. In response to a beta-3 agonist, we see that it is indeed phosphorylation is increased in wild type cells. And when we looked at knockout cells, that phosphorylation was dramatically increased. And when we looked at in this case, we looked at an immortalized cell line, a human cell line, which we added a RAL inhibitor to block the activity of RAL. We again saw an increase in the phosphorylation of the site. And this is just a quantitation of this data. So I'm not going to take you through all the data through which we understood the mechanism, but I'll summarize it in the slide. So what we found is that in the lean state, in which cells are catecholamine responsive, protein kinase A is activated with fasting, inactivated with feeding, but constantly being activated. This leads to the phosphorylation of DRP1 on this negative site, which blocks the binding of DRP1. And this keeps the dynamics of mitochondria in check, where you have equilibrium between the dissociation and association of DRP1. And so you lose the ability to fragment the mitochondria in these cells. Now, in the obese state, through a combination of hyperinsulinemia and resistance to catecholamines through other pathways, RAL is activated. And RAL constitutively interacts with DRP1 in a GTP-independent fashion. But when it's activated, it recruits the regulatory subunit of protein phosphatase 2A. And PP2A can come along and dephosphorylate this negative site on DRP1. And this results in the activation of DRP1, the chronic association of DRP1 with mitochondria, resulting in an increase of fission and an increase in fragmentation. So obesity through RAL activation and also through catecholamine resistance is associated with a shift towards mitochondrial fission, resulting in fragmentation of mitochondria in IWAT. So now, just to finish, we're still interested in this process and studying a few questions here, and we're very excited about trying to understand what is really driving RAL activity in obesity. And we don't really know the answer yet. I'll just tell you, we're looking at a few things. One, we're trying to understand the mechanism by which RAL mRNA expression is increased in obesity. We're trying to understand how RAL gap expression is decreased in obesity. And we're looking at other ways in which the RAL gap pathway is constitutively elevated. And everything that we're looking at now turns our attention to one factor, and that is hyperinsulinemia. So some of you may know that there's a movement right now to suggest that hyperinsulinemia per se in obesity is a primary factor that's leading to changes in all the metabolic abnormalities that that occur in obesity, including weight gain. So we're very interested in this idea now, and we're studying the role of hyperinsulinemia in changing gene expression and protein activities that lead to RAL activation. We're also asking this question about whether or not there are other factors involved in controlling the fragmentation of mitochondria. And I'll tell you that in addition to inhibition of RAL-GAP, there are also RAL-GAPs that are activated in response to insulin. And these are upstream G proteins like RAS and RAB10 that are activated in a G protein cascade through the recruitment of RAL-GAPs. So this is another way in which RAL could be activated, particularly in response to high-fat diet and hyperinsulinemia. I mentioned before, catecholamine resistance is also an important component of obesity, and this results in a decreased activity of the protein kinase A pathway. This also leads to decrease in the phosphorylation of this negative site and activation of DRP1. And then finally, there are other proteins in the mitochondria that seem to play an important role. And these are scaffolding proteins. One of these, ACAP1, is decreased tremendously in response to obesity. And this seems to play an important role in pKa-dependent activation or pKa-dependent inactivation of DRP1. And then there's another protein that we bumped into called MAVS. And I'll just tell you a little bit about this. MAVS is a scaffolding protein involved in inflammation that we've been studying for other reasons. It's a scaffolding protein in activation of the TBK1 pathway that Pong studied a long time ago. And it turns out that MAVS is dramatically downregulated in obesity. Both reduction in its expression and also the proteolysis of MAVS is increased in obesity. MAVS is negatively correlated with activation of TBK1. We used to think it was a scaffold for TBK1. Now we think it's a negative regulator. But it seems to play a role in DRP. Oh, I also point out that just like with RAL-GAPA2, MAVS is negatively correlated with obesity in humans. And this is like the data I showed you before. And when we knocked out MAVS in fat tissue, in fat cells, looked at IWAT, we see that although there's no effect of MAVS knockout on catecholamine sensitivity, looking at HSL phosphorylation, there's a pretty big reduction in DRP1 phosphorylation. So we think MAVS is somehow acting as a scaffold to support the phosphorylation of DRP1 and the negative regulation of this protein. And then thirdly, the other thing we're interested in is there an opportunity here for drug development? Can we use RAL inhibitors to treat obesity? And we're just starting to look at this. This is work from Wen-Min before she left the lab, and she's been giving mice RAL inhibitors. We see there's a small reduction in weight in these mice, and this is due purely to an increase in energy expenditure, kind of what you'd expect when you have a modest improvement in energy expenditure in obese mice. So we're now pursuing this line of investigation as well. So to summarize, let me just remind you that energy expenditure is repressed in obesity. We think this is due to a number of different reasons. There's inflammation in adipose tissue that's targeting a number of pathways. I didn't talk today about resistance to leptin. That could also be responsible for decreased energy expenditure. Catecholamine resistance as a consequence of inflammation and other pathways are also involved in repressing energy expenditure. And now we've added a new component, and that is the fragmentation of mitochondria, as I think another important component with respect to adapting to metabolic stress. So I'll just close there. Let me tell you about some of the people that have been involved in this work. We've done this work in collaboration with Joe Schoenenberg at UCSD, who's a mitochondrial expert. We've done a lot of the genomics work I told you about with Ron, in collaboration with Ron Evans. Michael Ryden at the Karolinska Institute has been involved with some of the human studies. The early work I told you about on RAL was done by Xiaowei Chang and Meiran Xiaowei Hung and Darryl Lito, and the work I told you about today on in vivo glucose homeostasis with RAL was done by Yulia Skoro-Kobatko, and the work on RAL knockout mice was done by Wen-Min Xie and a few other people in the lab. And I'll just stop there, and I'm happy to have a discussion and answer any questions for you. Thank you very much.
Thank you, Alan. Always very impressive presentations, and we learned a lot. So I think next section will host by Li-Heng Wang will lead the panelist discussion first, and then afterwards we will have the audience discussion questions.
Okay.
Yeah. Yeah, thank you, Alan, for the great talk. Giving us a very impressive talk about RAL1 function as a metabolic stress sensor in the adipose tissue to regulate glucose metabolism and also fatty acid oxidation through regulating the mitochondria fragmentation. So today we have 5 panelists. So the first panelist goes to Zhao Peng. Please unmute yourself.
Hello, Alan.
Fine.
Very nice, yeah, very nice talk as usual. So
Thank you.
it's a long story, but very super clear.
Thank you.
Yeah. So probably here I have several questions. Probably I should know better about some of them. So first one is does catecholamine and the PK signaling regulate RALA activity? I don't know that. I should know that the answer for that question. Yeah.
Yeah. So the answer to that is no. So it looks like catecholamines do not regulate RALA signaling, but RALA signaling may regulate catecholamine action. So
I see.
it's a little bit complicated, but we think RALA opposes the impact of catecholamines on DRP1 phosphorylation, but not other phosphorylations. Now, long term, it's a little different in brown fat. So all the things I talked about today with respect to regulating a DRP1 pathway with RALA is exclusively happening in white fat and is not happening in brown fat. And we do a brown fat knockout of RAL. We don't see these things in brown fat. But RAL is doing other things in brown fat besides glucose uptake. So it's a little more complicated. There is a modulation of catecholamine action through changes in the beta-3 receptor. And I didn't talk about it today. We're not quite ready to talk about that yet. So even though it was really complicated, what I presented was complicated. It may be even more complicated. Start looking at when we start introducing brown fat.
I see. So another question is that regarding the RALA mediates the interaction between the PB2A and the dephosphorylation of the DRP1. So is that pretty specific for the interaction just between PB2A and DRP1, or it may also mediate interaction between other substrates and the PB2A, because PB2A also negatively regulates the insulin signal in some, right? So I'm wondering, about that part.
Right. So I don't know. That's a really good question. I don't know the answer, and we haven't cataloged all the effects of that yet. So I would say that the reason that PP2A is targeting DRP1 is that DRP1 actually binds to RALA. Okay, so PP2, I don't know that we don't have any evidence that PP2A is activated by RAL. It's just that it's localized to RAL, and it's phosphorylating stuff that's already there in that complex. So that's why I think that we don't, for instance, we don't see a dephosphorylation of HSL because we don't think RAL is anywhere near the lipid droplet. But I have to say that we don't know where RAL is in this scenario. I kind of put it at the plasma membrane, but I don't really know that, and it could be in mitochondria. It could be in mitochondria. So it could be phosphorylating other things in mitochondria. But we know that DRP1 is constitutively interacting with RAL in a GTP independent manner. So it's binding by the way.
I see.
So it's a really good, I don't know, it's a really good question.
Yeah. So the last one is, the GWAS study showed the correlation between the MAVs and the MAVS, the MAVs with asthma. So, and it's related to TBK1. I'm wondering about amlexos, whether that protein also has some function mediating amylase effects.
Yeah, yeah.
That's
Yeah. So now we're getting into another area, but
yeah.
so we've also looked at MAVs and liver too, right? So
Uh-huh.
so MAVs is a platform for. I mean, MAVs is thought to be a platform for TBK1 action in
Yeah.
mitochondria.
Yeah.
And people looked at it macrophages and neutrophils and eosinophils, and it looks like it's regulating the danger signal process a little bit different from STING, but it's regulating the danger
Mhm.
signal process in these cells. But we don't see that in fat cells. So I think in fat cells, it's a negative regulator of TBK1 because it's
Mhm.
sequestering it away from STING. STING
Mhm.
is a bad guy. I mean, STING is upregulated. Oh, by the way,
Yeah.
MAVS is also upregulated in cold,
Okay.
where we have a decrease in inflammation. So I think MAVS is a negative regulator of TBK1 when it's in fat cells because it's sequestering TBK1 into mitochondria where it's less active. And
Mhm.
it's pulling it away from STING. When it binds to STING, it's super active, and
Mhm.
that's activating that cascade. So I think there's a tug of war between MAVs and STING.
Yeah. So in the liver, does it affect the interaction between TBK1 and the ACSL?
Yes,
The Jin Yang showed. Okay.
yes.
Got it. Okay. By the way, Wenmin is here as well in the
Oh,
audience.
okay, good. Well,
Thank
hopefully I
you.
did justice to her work. If I didn't, she can speak up and tell you.
Okay, thank you, Peng. Oh, Alan forgot to mention that today we reached a new record for the audience. We
Oh,
have up to 319 audience during
oh,
your talk. Yeah,
wow.
thank you very much.
Okay. Hopefully,
Yeah, so
I hope they stayed awake for all of it.
yeah, hopefully. Thank you. So the next panelist goes to Anyu. Aaron, please unmute yourself.
Oh, yes, Alan, great to see you here. Actually, I was a postdoc at Philip Scher's lab before I moved to UT Health. So I also work on mitochondria in white fat cells. So that's why I read your like Nature Metabolism paper about this story. It's a beautiful story, and again I learned a lot today. So maybe I have maybe two questions. I think one question is related to the fundamental difference between inguinal fat and visceral fat. So you show that the inguinal fat has more like fragmentation of mitochondria under high-fat diet, but actually visceral fat has a basal level of mitochondrial fragmentation. So can you tell us a little bit more about what's the mechanism driving this like basal fragmentation of mitochondria in visceral fat?
No, I can't tell you because I don't know. That's a really good question. So does it have to do with, it looks like RAL activity might be a little bit higher in EWAC basal, but
Uh-huh.
I don't know. I mean, I really don't know. And it could be that catecholamine action is different. It could be innervation of the visceral fat is different than subcutaneous fat. I mean, there's a lot of questions about, and like I said, there could be other factors involved too. So
Mm.
I think we've much to learn about this. I mean, I think people have looked at mitochondrial dynamics mostly in dividing cells and looking at its adaptation and energy needs in dividing cells, not so much in non-dividing cells. So I think we have a lot to learn. It's a really good question. We have a lot to learn about this.
Yeah. I think that's a kind of maybe a one explanation to show why like fat patterns are so heterogeneous. Maybe mitochondria is one of the major player here.
Yeah.
So my second question is about the broad fat, actually. You mentioned about the UCP1 Curie driven knockout didn't show a phenotype. So how about under cold stress? Do you notice actually there's more changes broad fat or there's changes in other like glucose metabolism?
Okay, so that's a really good question. And so there are changes with RAL under cold stress. but the changes have nothing to do with mitochondrial changes. So they mostly have to do with trafficking things that are going on. And one is GLUT4. It's very complicated, but as you probably know, cold stress, in addition to inducing a thermogenic response, induces lipolysis because of beta adrenergic receptors.
Yes.
And lipolysis gives rise to an increase in insulin, right? So in cold stress, there's actually an increase in glucose uptake in brown fat due to insulin. And RAL gap knockout mice, that glucose uptake is constitutively active. So in cold stress, there's actually more glucose oxidation in the RAL gap knockout mice in cold stress, and there's an improvement in cold tolerance. But this has nothing to do with mitochondrial changes.
Okay.
So it's all due to transport. Now, there's probably other cargoes that are also changing in response to RAL activation besides GLUT4 too. So I don't think we can explain everything by GLUT4. I think there are other changes, and there could be other transporters, or there could be receptors that are changing in response to RAL and changes in RAL and RAL exosystem interactions, which I didn't talk about today, and we don't know anything about that either.
Okay, thank you so much. I think that's all my questions. Look forward to see you in person, in some meetings. Okay.
Thank you, Anyu. And so our next panelist, Hannah, please unmute yourself.
Yeah, thank you, Lihe. Yeah, hello, Dr. Seychelles. Yeah, thanks for the talk. Learned a lot, not only scientifically, but also learned the positive attitude next time when I see negative data about that. Yeah, so maybe first of all, just a follow-up question of Dr. Anyu, because you were talking about the difference between IWAT and EWAT, right? But that is mainly based on the observation in mice. But in human, do you think that is the same? Because like at least for some single nuclear sequencing data, it shows that in human, those brown-like that fat mainly is present in the that visceral fat, but not in the subcutaneous one. So yeah, that's something I'm curious about.
Yeah, that's a really good question. I don't know. I mean, I think, it's really hard to do this in humans because you have to do so many to do a substantial study where we really understand that we need to have a big population of humans. I mean, look, it's hard to do it in mice, right? I mean, the heterogeneity just in black 6 mice, brothers and sisters in the same cages, in the same environment, with the microbiome being the same, put them on high fat diet, and they gain different amounts of weight, right? And some of them don't even gain weight at all, and they're genetically identical, and they have the temperatures are the same. I mean, so who understands that? I mean, so there's a lot of heterogeneity, and when we start looking at people, oh my God, there's so much heterogeneity. So I think we would really need to study big populations. I don't think we have the tools yet to do this, to really look at characteristics of many cells of many, many, many people. I think this is going to take some more time. It's not the same as doing a GWAS study where you get a little spit or some blood. This is a little bit harder.
Mm-hmm. Yeah, indeed.
So anyway, this is a fancy way of me saying I have no idea. Okay.
Okay. Yeah. Learn again. Okay. Thanks. Yeah. So another question is that you mentioned that you observe in this RAL-A knockout mice, you see let me check. That's a mitochondria elongation, right? And then that accompanied by increased fatty acid oxidation. So what is the link between these two observations?
I think that the longer mitochondria have higher activity, are more active.
Okay.
And when we look at this in a mitochondrial, so when we do these seahorse studies in isolated mitochondria, as opposed to in isolated cells, we see the same thing. We see increased activity. So I think that the longer mitochondria are more active for whatever reason. It could be I really don't understand the biophysics of that, but they seem to be more active.
So have you checked like those key enzyme like CPT1A, 1B, or other OXPHOS proteins, whether they are
Yeah,
different?
we don't really see a difference in those proteins.
Okay, nice.
Yeah, I mean the overall numbers are different, and if you go from tissue to tissue, but we don't in isolated mitochondria, we don't really see that much of a difference. So I think we still have to understand, we still have to do more to understand that better.
Yeah, that would be interesting. Yeah, and another thing, maybe I missed that. So have you checked the like UCP1 expression in this wild type and knockout mice, whether they are different or not?
They're not different.
They're not different.
Yeah,
Oh.
and there's no difference in Beijing.
Okay, because otherwise I'm thinking maybe there's an increase in fatty acid oxidation. So what is the driven force for that? Maybe there's an increase that uncoupling or something that can also be a possible reason for the increased fatty acid oxidation.
I mean, we've just done, we've just done the basic experiments on this, and there are going to be differences. So, for example, ACAP1 and MAVs are dramatically upregulated in cold exposure. So there are going to be differences, but I think we just don't know if it's dependent on RAL or not. We're just beginning to investigate whether RAL is changing in cold exposure and sympathetic activation in terms of its expression and stuff.
Yeah, that just reminds me another thing is that although UCP1 is not altered, whether did you have a chance to look at those UCP1 independent sabogenesis in the mitochondria or in the cell? Yeah, maybe that that may be also possible. Yeah.
Yes, that's a very good point. We haven't looked at that. Ask Pong about that because he did a little bit of that when he was in my lab. Looked at the creatine cycle, but we haven't done that.
Okay, yeah, that's because now at least in human there are some evidence that UCP1 independent sabogenesis might be quite important.
Yes.
Yeah. And maybe a last one is just to seek your opinion because you mentioned maybe this inhibition of RAL-A, right, is that therapeutically have some translational value, at least by increasing fatty acid oxidation. So do you think it is possible that in addition to just enhancing this energy expenditure and those all enhance the fatty acid burnout, whether it has some other benefits? Because I used to read a paper saying that enhanced fatty acid oxidation, it will theoretically increase those acetyl-CoA level within the cell, right? Maybe it has some other like epigenetic effects on the cells itself.
Yes, I mean it's a good point, and it's just that we're just at the beginning of this. I mean, I would say right now I'm a little more concerned with identifying the liabilities of RAL inhibition because of what might happen in other tissues. We have a big story on this in liver now, so there's a whole nother story with RAL and RAL gap in liver, and but that's not even beginning to look at other possibilities. RAL inhibitors were originally developed for cancer because RAL is an effector of RAS, so that's why people were originally looking at it. But Jesus, there's so many other things that RAL could be doing, and RAL, the whole body knockout of RAL is lethal, and so I can't even begin to get into this. I just don't know. That's why we have to do experiments.
Yeah, okay. Yeah, thank you. Very nice to talk to you.
Thank you.
Yeah, that's all my questions. Thank you.
Thank you, Hannah, for the great questions. Actually, follow Anna's question. I have one question. Alan, have you ever looked at like I'm not sure whether other people have looked at the GLP-1 receptor agonist after its treatment, whether RAL-A's level is changed in the adipose tissue, and plus, like because we saw very minor effect from the RAL1 inhibitor, is it possible to combine the treatment with RAL1 inhibitor with like GLP-1 agonist?
Yeah, did you read my grant? I mean, that's exactly right. I mean,
No, I didn't.
That's exactly what we need to do. So yes, it's a very good idea, and we haven't done that yet. So
I see.
we're starting to look at that. Yeah. I mean, my philosophy about this, until we know it better, is that RAL inhibition or any other mechanism to increase energy expenditure alone will not be very effective without some kind of reduction in food intake,
Right.
which could include GLP-1 drugs or diet. But as we all know, diets that don't work. So to me it makes sense that doing these combination experiments together will be very interesting and maybe able to reduce some of the liabilities with the GLP-1 drugs, especially the muscle wasting, and maybe even nausea too. So that's a really good idea. I think a lot of people are thinking about that. We're thinking about that with other targets that we have too with energy expenditure.
Okay, great. So then welcome our next panelist, Hongxue. Please unmute yourself. Oh, Mengxi.
All right. Nice talk, Alan. So I'm Hongxue in Aritabas lab at Columbia University. So we did, metabolic dysfunction associated steatohepatitis MASH and atherosclerosis. So I purely focus on the MASH. So maybe you already mentioned some, you already did something in the liver disease. So can you give us a little bit more information about the function of the RAL in the liver disease, like the method, the MASH, and also even the MASH HCC?
Yeah, really good question and really complicated answer. So I'll say a couple of things. One is, again, this is confusing, but I'll say that when we knock out RAL and white fat cells, like I showed you, there's a resolution of steatosis. And I think this is an indirect effect of oxidizing more fat in white fat cells. So I think it's purely an indirect effect of that. And then parenthetically, when we knock out RAL gap in brown fat cells to increase glucose uptake, we also see a resolution of steatosis. Again, this is one of those confusing things. We have the opposite things, but they're giving us the same impact. In that case, I think because it's in brown fat, it's because of the glucose uptake going up and the insulin levels coming down. So I think it's an effect of reducing insulin levels, but I'm not sure about that. So we're looking at that carefully now, and we see changes in lipogenic gene expression in the liver that's consistent with a reduction of mTOR activity because of a reduction in insulin. So I think it's all consistent with that, but it's not proven yet. So there could be something else going on. Now, we're also looking at RAL in liver too, in hepatocytes, and there it's really interesting, but it looks like RAL is regulating the trafficking of some other proteins, particularly the LDL receptor. And we're very far into this now, trying to figure out the mechanism, the impact of RAL, looking mostly at cholesterol metabolism. But of course, as you know, cholesterol content in liver has a big impact on steatosis because it's so toxic. So there's lots of ways where RAL could be regulating liver, directly and indirectly.
All right, thank you.
So, Hongxue, do you have more questions?
No, that's all. Thank you.
Okay, thank you. So then go to our next panel, the last panelist, Mengxi. Please unmute yourself.
Thank you, Li Heng. Hi, Dr. Satel. Really enjoy your talk. I have some questions regarding RAL-A and its role in glucose homeostasis. You mentioned that both activation and knockout of RAL-A seem to improve glucose tolerance. So how do you reconcile these findings mechanistically? And given this dual effect, do you think RAL-A can be developed as a therapeutic target for metabolic diseases? And also, what is the overall effect of RAL-A inhibitor on systemic glucose tolerance or insulin sensitivity in vivo? Thank you.
Yes, thank you. It's a very good question. It's something we're struggling with a lot. So again, I'll point out that RAL activation in white fat doesn't do anything to glucose uptake. In brown fat, it really increases it a lot at the basal level, even in the absence of insulin. because it's mimicking insulin action, because insulin activates RAL. Okay, so in brown fat, you get a big increase in RAL activity. Okay, and you get improvement in glucose homeostasis. So that's why we expected to see the opposite in the RAL knockout. But I think because RAL in white fat is controlling energy metabolism, the change in energy metabolism trumps the change in glucose uptake. So when you lose weight, because you have more fatty acid oxidation, you improve glucose uptake indirectly. So that's why the glucose homeostasis improvement in animals in RAL knockout mice, it's just like adding a beta agonist to fat cells. It's counterintuitive because beta agonists oppose the action of insulin. But if you give chronic CL to mice, you see a big improvement in glucose homeostasis, right? Because it's more than just weight loss. It's because you're increasing the metabolism of glucose in the fat cell. So it's like the effect on energy homeostasis trumps the effect on glucose uptake to improve the glucose homeostasis. That's a little hand, excuse me, a little hand waving, but that's my interpretation of the data. Excuse me.
Okay, thank you. Then that's all my questions.
Thank you, Mengxi. Thank you to all the panelists. So now goes to our audience. So welcome audience to ask your question. So if you have question, please raise your hand or post it in the chat box. So our first audience question is from Candice. Are you there? If you are there, please unmute yourself. Candice, are you there? So if not, I'm going to read the question for Alan. So Candice said, very fascinating talk, very informative. I wonder if the RA regulation on mitochondria fission and fragmentation is also through organelle interaction, such as ER mitochondria, lysosome mitochondria interaction by the GTPase function.
Yeah, very good question. I don't know the answer. We're looking at this right now. We've developed a probe for RAL-A in collaboration with Jin Zhang at UCSD, and we're really interested in trying to figure out where this is all happening. We don't know. We just do not know where this is happening. So it could be happening at the mitochondria, could be happening in the ER, could be happening in MAM, could be happening at the interface of these things. It could be regulation of localization of DRP1 or RAL itself or RAL gap. I mean, I don't know. Who knows? I mean, so there's a lot of things that could be happening. So that's a very important question. I don't have the answer to.
Yeah, following this question, I just wonder, does RAL-A directly involved in regulate the lipid transport in the cytoplasma?
Yeah.
Between, the mitochondria.
Yeah, so Yulia Scorgobato has been trying to figure out if RAL-A is controlling the trafficking of the fatty acid transporter. This is a very difficult field that has killed a lot of careers, and we've been working with Andreas Stahl to try to figure out if this is happening. You may know that insulin. There've been a lot of papers over the years showing that insulin increases the fatty acid uptake, and controversy about whether it's a receptor-dependent thing or not. So I would say that we do, so in our RAL gap knockout mice, when we've done proteomics on plasma membrane fractions from the mice, we do see an enrichment in the plasma membrane of some of the fatty acid transporters. So it's possible that fatty acid uptake is increased. Likewise, it could also be possible that fatty acid transesterification to TGs could be increased through other mechanisms. And some of those enzymes could be regulated. So we have a lot to learn about this, and these are really hard studies to do, and that's why that's one of the reasons we haven't made progress.
Okay.
Yeah.
hopefully the new techniques can help. So, Julie,
Yeah,
do you have a question?
take advantage and just follow up with your talking about the the MAVs or STING, STING signaling, because mitochondria is such important for both pathways, including the TBK1, that's the master proteins in your lab. I'm just wondering whether the RA can regulate, either the how to incorporate whether activate MAVs or STING-induced, inflammation pathway. Have you ever done that RAP-A in the regulation of mitochondria, and as well as I think in the STING field, they also reported in the adipose tissue, especially there's a report regarding the STING's regulation on the polyunsaturated fatty acid. Have you ever perform, or do you have any perspective for this part?
Yeah, we've done quite a lot to explore this, and I would say that I have no perspective on this because yes, it looks like under certain conditions you can get activation of inflammatory pathways, especially junk, with RAL activation, and there are reports of RAL being related to NF-kappa B from Mike White, old studies from Mike White. There's also reports that IKK epsilon binds to the exocyst proteins, especially SEC5. And so there's a lot on this with respect to exocyst and regulation of phosphorylation and mitochondria. And I would say that we've been looking at this for like 10 years, and I'm just as confused now as I was at the beginning. So it's very hard to know what's cell autonomous and what's not in this area. But I can't rule out that there's an impact of the RAL pathway on MAVs and STING regulation of TBK1 or regulation of IKKE, and IKKE regulates PDE3B. So there's a lot of opportunities to get confused.
Thank you.
And I remain confused.
Okay, so our next question go to Zeqiu. So Zeqiu, please unmute yourself.
Thank you so much for your talk. And my question is, could you comment on the insulin resistance in obesity while the mice are hyperinsulinemia? Because based on your talk, it seems like that insulin resistance is not only occurring, it's not occurring at the insulin receptor binding site, since the RAL is still activated. So what is the counter insulin action in this case, since you also mentioned about the catecholamine resistance? And I wonder whether glucagon could play a role, and I wonder what's the evidence of catecholamine resistance came out. And I personally use isoparental CL, and I find they're not as good as norepinephrine just by itself. So could you comment on that? Thank you.
Yeah. This is like another seminar. Okay, you're asking a question I'm very interested in, and there's several people in my lab working on this question. So it's almost like another seminar. But we think that hyperinsulinemia is a trigger for inflammation. So we think that, I think it's very hard to know if hyperinsulinemia precedes any of the sequelae of events of obesity, insulin resistance, catecholamine resistance, all the changes in metabolism and in different tissues, and the interaction of the liver and fat cell. There's also a big story about the degradation of insulin in liver that may precede some of the events that are, to increase insulin action, insulin levels that may precede some of the effects of obesity. And likewise, elevations in basal lipolysis that June Liu has been looking at with the G0S2 gene may also precede some of the effects and even lead to hyperinsulinemia because of the lack of inhibition of ATGL. So there's a lot of stuff going on here. And I don't, like I said, it's another seminar to talk about this in detail. We do have a lot of data on this right now, but I think to me, I'm really I'm much more open to this idea that hyperinsulinemia it could play a primary role in all these bad things that happen in obesity.
I see. Cool.
I
Thank you so much.
know I'm not answering your question, but
Yeah.
it's too much. There's just too much there to answer.
I see. Yeah. I'm still a PhD student, so I'm still thinking about this question a lot. Like, what is exactly insulin resistant? And I find there's many
Yeah.
levels, maybe at the
Yeah.
insulin receptor level, maybe in the different signaling pathway interaction. Yeah.
Yeah.
But I also have one more suggestion on the model you're talking about. The model you showed for stress is just using cold and also fasting. But I personally study acute stress, which I restrain the mouse to put the mouse into fight and flight response. And I
Yeah.
find that's a really beautiful state because you have normal insulin level and the acutely induced catecholamine secretion and also glucagon's action. And I think there's more biology in that part. Maybe it's also a model you can use or think of. Thank you so much.
Yes, I just want to say that's a really interesting model. So we're looking at things like that. We're thinking a lot about glucagon, more from the liver than from the fat. I know there's some receptors in the fat cell, but glucagon and glycogen are very important
Yes.
controllers of metabolism. And we have a paper coming out in the JCI in the next couple of days, I think, on glucagon and starvation with respect to gluconeogenesis. So you keep your eye out for. I won't talk about it because that'll take me all night. But
Yeah,
and
I'm really excited.
I want to get to my glass of wine, but there's something there, that's kind of interesting too. And glucagon resistance happens in obesity.
Yes. Yeah, I study glycogenolysis, so it's the acute state of glucose release from liver, and I find the glucagon action is really dramatic and overruns insulin signaling like really strongly. Yeah,
Yeah.
but thank you so much.
Yeah, thank you, Zeqiu. Actually, Zeqiu is from Gokhan's lab. So, our next question go to Wen. Please, unmute yourself.
Oh, hi, Alan, it's me.
Hi, Wenmin. How are you?
How are you doing?
Good.
Yeah, actually, one of my question goes for, first of all, very great talk. It's actually my first time to hear you talk from an outside angle. So I can really tell you organize your research very well, from the inflammation of glycogen to insulin signaling. So I actually, I think you are one of the best person to ask about what's the relationship between inflammation and insulin resistance?
Yeah, that's a really good question. So first of all, I think you meant to say that I did a good job organizing your data. Okay, so that's the first thing I want to say. So I myself, just to answer this question with a short, precise thing, I think that inflammation causes repression of energy expenditure, which causes insulin resistance. So I think that back in the early 2000s, when everybody was trying to find pathways, including me, trying to find pathways in insulin action that were repressed by inflammatory signaling, either through junk or through IKKs or stuff like that. Every time we looked at upstream signaling and insulin action, like looking at IRS phosphorylation or AKT, and try to find sites that could be phosphorylated by junk or by some of these kinases, and we did a knock-in mutation, it never had any impact. So, and Morris White did this. He made tons of mice, and they never had any effect. There was never a phenotype when he blocked the inflammatory signaling, and with TNF or anything like that. So there's no doubt that if you give fat cells TNF, they de-differentiate and become less responsive to insulin. That's a program that they follow. But I think in vivo, I think all the effects of inflammation are to repress energy expenditure. And when you repress energy expenditure, you make cells less sensitive to insulin. So I think it's indirect. I think it's true, like, improving insulin sensitivity with beta agonists is the same thing. When you burn some energy, you make cells more sensitive to insulin. So that's the opposite effect.
Okay, so would you think like pressing inflammation would be a good treatment for insulin resistance?
Yeah, I think repressing inflammation is going to help insulin resistance. And I think that, we've shown this with Amlexanox, right? So that's one way you're pressing one of the effectors of inflammation and you dramatically improve insulin sensitivity, but it's an indirect effect. Because if you give it directly to cells and look at insulin sensitivity, it doesn't improve insulin sensitivity directly. It's an indirect effect of the burning of fat.
Okay, thank you very much.
That's my view. I mean, everybody doesn't agree with that view. I think always, my colleague Jerry Olesky really thinks that inflammation is directly repressing insulin sensitivity through EVs and stuff, and I just don't. I see it in a different way. Since he's not here, I'm happy to say that he's wrong.
Okay, I hope I can see you sometime this year.
Yes.
Okay, thank you. Have a good time.
Yeah, thank you, Wen. So our next question goes to Xiangqing He. Please unmute yourself.
Okay, thank you. Very, very interesting work. So I'm wondering that with the REL-A regulate mitochondrial dynamic in other cell types, or if this regulation is specific to adipocyte and hypophyte, because mitochondrial dynamic is seems like a common phenomenon in other cell types. So that's my question. Thank you.
So is your question, is it happening in other cells? Is that what was
Yeah, yes.
Yeah, it probably is. I don't know. I mean, we haven't looked. We've only looked at fat cells.
Okay.
So
you only detect in the adipocyte.
We've only looked in the adipocytes. So we haven't looked in other cells yet.
Okay, I see.
So Alan, I think we finished all the questions from the audience. Actually, follow Wen's question. I just came up. I just wonder for the PPAR gamma agonist, like the TZD drugs, how does that going to impact RAL-A activity in the adipose tissue?
Yeah.
Yeah, it functions in a different mechanism.
Yeah, I think that it's very confusing to me, actually. I think, with PPR gamma activators, you have many different things going on. I mean, you have increase in adipogenesis, and that, I don't know what that's going to do to this pathway. I mean, I just have no idea. You have a reduction in inflammation through inhibiting the TNF pathway, and that's going to have another effect. But then you have upregulation of a lot of anabolic genes in fat cells, including some that cause catecholamine resistance. And then you have activation of genes that are involved in the thermogenic response in mice anyway. I don't think anybody's seen that in humans, but I think that's something that we see in mice. So there's so many different things that PPR gamma activators are doing, and they're doing different things in pre-adipocytes and adipocytes. So it's really hard to sort this out. And my lab's been working on this off and on for 30 years, really, and I think we still don't really know, because all the agonists are different. They all induce a different gene expression pattern, and there's some partial agonists and full agonists and inverse agonists. And so I think, and then knockouts are very difficult to study because they block adipogenesis, right? So I think it's really tough even though we've been working on it. It's not the centerpiece of my lab. I was working on the discovery of the TZDs back in the early days. So that's when I was working on it. But we found some genes that were sensitive to PPR gamma, like CAP, and other genes. So I don't really know. I really don't feel like I can answer that question. It's a really good question.
Okay, thank you. Okay, thank you, Alan, for your great talk today, and also thank you to all the panelists for joining us today. To make a great discussion after Alan's talk, and also thanks to all the audience today. See you next week. So next week we invite Sida Faruqi from Oxford University. So she's going to talk about genetics of obesity. So hope you guys can join us again next week. Thank you, Alan.
Thank
Yeah,
you.
have a very nice weekend.
Thank you. Thank you, everybody. Great seeing you.
Yeah, just asking me to say hello to you, and he is in the
Oh, that's nice.
travel. So yeah, just say hello to you.
Okay, great. Thank you. Thank you so much.
Thank
All
you.
right. We'll see everybody.
Thank you.
Bye-bye.
Bye bye.
Thank you. Bye. Have a nice weekend, everyone.
You too.